longer, and we're going to be taking a look at our last portion of the nervous system, where we look at different types of receptors as well as our general and special senses. So for sensory receptors, and we have a number of different ones around the body, and their main purpose is to respond to what we would call a stimulus, okay? Anything that can trigger the receptor to respond. And we have sensory receptors not just on the surface of our bodies, but also internally so that we can maintain homeostasis. And each receptor is specific to the type of stimulus that it's going to respond to. For example, we have photoreceptors in our eyes that respond to light energy. We have different types of receptors called hair cells in our ears, which respond to sound waves. So everything is going to be kind of different. And if you remember before, when we were in our earlier part of the unit, we have what we call ion-gated channels. They might be called voltage-gated ion channels, where basically we know that we are going to create electrical potentials to send signals down these nerves using these ion channels. And if you remember, and you should remember because you're going to need to know this for your exam, we are focusing on specifically the ions sodium, potassium, and calcium. And essentially, these sensory receptors are what we call transducers that convert stimulus energy into electrical energy simply by changing the resting membrane potential, right? Where we're opening these ion channels, and either we create an action potential to send a signal or we don't. So there are two categories that we can put these receptors into. We have general sense ones and we have special sense ones. General sense ones are simple and they're found all over the body. And we have ones that we would categorize as somatic, right, where we are under specific control, conscious control. This could be receptors in the skin or in muscles, joints, tendons. And then we have visceral receptors, which are found in internal organs, and they respond to a number of different changes internally. Those are general senses, which we'll take a look at soon. And then our other category, which we'll look at the latter half of this talk, will be special senses. And these are specialized receptors. They look very differently than our general sense ones, and they are comprised in the sense organs inside our head. And so our special senses are termed olfaction, associated with the nose, gustation, associated with the tongue, vision, associated with the eyes, audition or auditory response, hearing, and equilibrium, associated with the ears. So if we want to put these receptors into different categories, okay, and these categories could fall into either general or special senses, depending on what the receptor is. We have certain ones that are found on the external surface of the body called exteroceptors, okay, we have that word external in it. We have ones that are found on the internal organs of the body called interoceptors. Sometimes in other texts, you'll see them called visceroceptors. And then we have this last category associated with muscles, tendons, and joints called proprioceptors. If we want to look at additional categories that we could place some of these receptors into, okay, not just by location, but by what they respond to, we have some additional ones. Chemoreceptors being one of them. And chemoreceptors can be exteroreceptors if they're detecting things on the outside of the body, but they can also be internal. And all they do is detect changes in chemicals. So they detect chemicals dissolved in fluid. Okay, so this could be in the blood. This could be in something you taste, something you smell, those types of things. Chemoreceptors respond to chemicals. Thermoreceptors respond to changes in temperature. And you could see this, you could think of that first word thermo, thinking of a thermometer. And we see these present in the external surface of the skin, but also in our brain and the hypothalamus, which controls our internal body temperature. Photoreceptors are exclusively found within the eye. These are special sense photoreceptors, and they respond to changes in light. Mechanoreceptors are found all over the body, and they could be categorized in a number of different areas as exteroreceptors, interoreceptors, or even proprioceptors. And they respond to touch, changes in pressure, vibration, and stretch. Essentially, anything you're applying mechanical energy to are what mechanoreceptors are going to feel. They detect distortion. The last category is nociceptors, also called pain receptors, and we do have them both externally and internally. For our tactile receptors, and mostly we think about mechanoreceptors as part of tactile or sense of touch, where we can feel changes in stretch, pressure, vibration. And we have a few categories for these. We can consider them encapsulated or unencapsulated. Um, Things that are considered unencapsulated are free nerve endings. And the ones that are encapsulated are usually tactile corpuscles, also called Meissner's corpuscles. And then the other ones are called lamellated corpuscles, also called Pacinian corpuscles, which you can see highlighted in this image of the skin. And these are things you had to learn previously on your previous lab exam. So again, here are a few of those tactile receptors without pictures. Free nerve endings, okay, are unencapsulated. They're very simple tactile receptors. They can feel pain or temperature changes as well as touch and pressure. We also have some tactile receptors in our hair. So they're found in the root hair plexuses, and they can detect movements in hair. So when you distort one of your hair follicles, this is what is detecting that movement. These are considered unencapsulated, so they just don't have a capsule around the receptor. The last one are these tactile discs, also comprised with tactile cells or Merkel cells, something we also learned in the skin, present in the epidermis, so the epithelial layer of the skin, and they do respond to light touch as well. These are all unencapsulated. They're specific cell types or free nerve endings that are just going to detect mostly just light touch or distortion movement. Our encapsulated ones, we have a few of them. Do not worry about the Krauss bulbs. I'm not using those, but we will revisit these lamellated Pacinian corpuscles, okay? These are encapsulated. They have multiple layers. They are found deep in the dermis, even in the hypodermis, and they feel deep pressure, coarse touch, so heavy touch, and high frequency vibration. So since they're deeper in the skin, they're in the deepest layer of the dermis and even into the hypodermis, they are going to feel deep pressure, heavy vibrations, unlike the ones we were just looking at, which feel light touch. A few other ones, do not worry about the Ruffini corpuscles as well, but the other one we care about that's encapsulated are these tactile or Meissner's corpuscles. These are in the area of the dermal papillae, so they're a little bit higher up in the dermis than those Pacinian corpuscles, and they feel light touch and they can recognize changes in pressure or shape for objects that touch the skin. So they are for light touch, but they're discriminative, so we can actually recognize what is touching us. Those are the tactile Meissner's corpuscles. If we move into away from tactile receptors into proprioceptors, okay, which mostly feel stretch, okay, they're specialized mechanoreceptors, right? They feel mechanical energy. These are only found in muscles, tendons, and joints, sometimes in internal organs, but we're going to focus on these three types of proprioceptors. Muscle spindles detect stretch in muscle. They have the word muscle in them. Golgi tendon organs detect stretch in tendons. They have the word tendon in them. Use that as a study aid. And then joint kinesthetic receptors feel stretch in articular capsules, which are making up our joints, okay? So they feel stretch within the joints, all of which have the name of what they detect stretch in the name of the proprioceptor. These are just what they look like. We mentioned this before when we looked at those dermatomes where you have what we call referred pain, right? Where you might feel pain in your heart. You might be having a heart attack, but you actually feel that pain manifest in the skin on the left side of your body. This is referred pain, which is termed inaccurate localization of sensory signals, meaning even though you are feeling pain in one part of the body, it will manifest in certain sensory parts of the skin in another. This is just to review referred pain. Remember, it's in your previous chapter when we looked at dermatomes. We're going to move into special senses. The first one we're going to look at is olfaction, your sense of smell. And olfaction is detection of odorants dissolved in air. And those odorants are simply chemicals, which tells us that the receptor we're going to look at are chemoreceptors, detecting changes in odorants or chemicals that come in contact with our nose. We have an olfactory epithelium, which is where we're going to have those chemoreceptors located. So we have those olfactory receptor cells, which detect the odorants. And then we have two other types of cells, which basically keep these receptor cells from getting damaged. Again, the main focus is these olfactory receptor cells, which have the receptors, those chemoreceptors, to detect odorants. This is kind of what that looks like. Again, we're focusing in here on those olfactory receptor cells. We have these olfactory hairs, which are the chemoreceptors that detect odorant molecules, okay, our chemicals, and they're going to send the signal up to the olfactory nerve. Our second sense is gustation, your sense of taste, okay? If you remember when we were looking at lobe associations, insula, that lobe that's very deep underneath the other lobes, is the one that senses taste. And we have cells which have chemoreceptors as well because we are taking chemicals into our mouth to detect. Those chemoreceptors are present on the taste buds. And we call our essentially those cells gustatory cells for gustation. We know that the taste buds exist in the papillae of the tongue. These are different raised areas of the tongue, which either contain taste buds or don't. And some of them play a role in breaking down food, such as the filiform ones, which are short and spiked, which help us manipulate our food, meaning help us tear apart our food, but have no role in taste. Oftentimes, if you get licked by a cat, I have cats, right? They have a very rough tongue. This is because most of their tongue is this filiform papillae, which has no purpose for taste, but helps them break apart food. Our next papillae is fungiform papillae. Fungiform meaning fungi, they're mushroom shaped, okay? They contain a few taste buds, so they're not our primary area of taste, but they do have taste buds present with those gustatory cells, chemoreceptors, and these are located on the tip and sides of the tongue. Our other two are foliate. They look like leaf-like ridges, foliate meaning foliage for plants. They only have a few taste buds as well, only during early childhood, and they're located on the posterior lateral sides of the tongue. And then the last ones, which have the most taste buds, are our valate, also called circumvallate papillae. They're very large and circular. They have multiple rows of which are on the posterior dorsal surface of the tongue. This is where we primarily do our taste. And again, here's a picture. You can look at each of these. And as far as our sense of taste goes, we have supporting cells and basal cells, just like we would in the nose, okay? But what we care about is the receptors, which are our gustatory cells. Again, we just did smell and taste very, very briefly, and each of these have chemoreceptors, right, detecting chemical changes. The eyes and the ears are much more complicated. Okay, so that's where we have the other half of this PowerPoint. We do know a few features about the eye, right? And what I want you to focus on is the presence of a posterior and anterior cavity for the eye. We have a posterior cavity, which is the largest portion of the eye sitting behind the lens, and it contains our vitreous humor, a jelly-like substance that keeps the eye shape together. And then in the front of the lens, we have an anterior cavity, which has our aqueous humor. And aqueous humor is kind of sitting behind the cornea, okay, so in the front of the eye. Again, so named posterior and anterior for their locations. For the eye itself, we have a number of different tunics or layers that are present that we're going to look at. And you can kind of see in this picture simplified, we're going to look at an outer layer containing sclera and cornea, a middle layer, which we call the vascular tunic, containing choroid, ciliary body, and iris. We have the lens, And then our innermost layer, we have our retina, which contains a number of different layers, which we separate into categories of pigmented and neural. And again, this is just showing a lot more features to those structures. For the outer layer of the eye, which we term the fibrous tunic, we have the sclera, which is the white of the eye, and we have the cornea, which is a transparent window into the eye. Again, the cornea is going to have that aqueous humor behind it, and these are portions of the cornea is a portion of the anterior cavity. The vascular tunic contains many blood vessels, which is why it's termed vascular. The main area full of capillaries and blood vessels is choroid, and that is going to bring nourishment to the retinal cells. We also have a structure called ciliary body, consistent of ciliary muscles and processes, which help hold the lens in place and help for its manipulation. The iris is also part of this. The iris is the color of your eye. It is also contains a number of smooth muscle, which can manipulate the pupil, the hole in your iris, where you can allow certain light to go through, right? So that it bounces off the lens onto your retina. The pupil is controlled by a number of different muscles, and there is even a reflex associated with it when light comes into the eye. This is showing those muscles. The retina is the innermost layer of the eye. It contains a number of different cell layers, okay? We have our photoreceptor layer containing rods and cones, which are our photoreceptors, which we'll take a look at. We have bipolar cells here, which are present, connecting with those rods and cones. We have a ganglion cell layer, which is the innermost layer. In some of my classes, we do every single layer of the retina. There are several, but for this class, we're just looking at some small sublayers. The ganglion one being the innermost one that is actually going to send those signals to the nerve fibers of the optic nerve. Don't worry about the interneurons here. This is kind of what it looks like. There's obviously more layers present in here, but we're just focusing on the photoreceptors with the rods and cones, the bipolar cells here, and then the ganglion cells that send that signal to optic nerve. There are a few other structures present on the retina, aside from its layers that make it up. We have optic disc, which is where your optic nerve connects to the retina. It is considered your blind spot. We have macula lutea, which has a center of cones called fovea centralis, cones being photoreceptors that look for that are present for bright light and help us with color vision as well as our sharpest area of vision. Surrounding macula lutea and fovea centralis, the rest of the retina is primarily rods, which function in low light settings. They allow us to see image outlines as well as grayscale color. So our photoreceptors, rods and cones, you won't have to identify them by their shape, okay? We have more rods than we do cones because we know that cones are kind of concentrated in fovea centralis in the center of macula lutea. Rods are found in the periphery of the retina. They respond to dim light, okay? And they are going to give us blurry images as well as grayscale images. Cones only respond to higher light, bright light. They allow for color vision as well as our sharpest vision. This is just kind of what they look like. And those boxes are very good for kind of showing the differences between rods and cones, the boxes with descriptions next to this picture. We'll move into the ear to finish. So the ear is another more complex structure than the others. We have three different sections of the ear, our external, middle, and internal ear, which we'll take a look at. The external ear is consistent of three structures, your auricle, your external acoustic meatus, and your tympanic membrane. We'll take a look at a picture of that. Auricle or penna is also a name for it, is the outer portion of your ear. Your external acoustic meatus is your ear canal, which contains a number of ceruminous glands which produce cerumen or earwax. And your tympanic membrane is your eardrum. Essentially, these are funnels. Even the tympanic membrane, your eardrum, is funnel shaped to move sound waves to the middle and internal portions of the ear. So again, this is the outer portion, auricle or penna, external acoustic meatus, eardrum, tympanic membrane. Sound waves travel through here, vibrate this tympanic membrane, which in turn vibrates these three bones within the middle ear. The middle ear is an air-filled cavity called tympanic cavity, and it contains a few openings, one of which is to your auditory tube or your eustachian tube. It allows the release of fluid as well as air when pressure builds up in there. So if you've ever been on an airplane and you're at a higher altitude and you need to swallow to pop your ears, you're opening this auditory or eustachian tube to do that. The three bones, also termed ossicles, small bones that are found within the middle ear, are malleus, incus, and stapes, and they will vibrate in that order. Malleus resembles a hammer or a mallet, incus resembles an anvil, and stapes resembles a staple or a saddle stirrup. And they are all going to vibrate with those sound waves against an opening called the oval window. And the oval window is going to vibrate the fluid within the inner ear. Do not worry about these two muscles that are attached to the ossicles. Here we have a picture, malleus, incus, and stapes, which will vibrate. You can see the eardrum right here. They'll vibrate against, again, this oval window here, leading us to the inner ear, which is filled with fluid. The inner ear is comprised of two labyrinths. We have a bony one, which is hard on the outside, filled with its own fluid called perilymph. And we have an internal one called the membranous labyrinth, filled with its own fluid called endolymph. Both of these fluids are going to vibrate, okay, and respond to those sound waves. The three internal ear structures that these labyrinths line are cochlea, which is used for hearing, vestibule and semicircular canals, which are used for equilibrium. You can see kind of the outer and inner membranes just lining these structures. This is cochlea, the snail shell in your ear. This is vestibule, and then these kind of canal structures are the semicircular canals. That is all you do need to know for these guys. If you went into a more advanced A&P course, we would go a lot more in depth into their functions and how they function for hearing, as well as additional things for the eyes, nose, and mouth. But for now, those are the basics that you do need to know, along with the rest of the nervous system material. So that is it. Please let me know if you have any questions via email, and good luck studying for your lab exam this week.